This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 80 for April 30th, 2010. Hi, everybody. I'm Vincent Racaniello, and this is TWiV, the podcast where we talk about the week's news in virology. Joining me to do that today from Western MA is Alan Dove.
Good to be back.
Welcome back. Good weather today out there?
Absolutely gorgeous out here today, finally.
Good. Here too. It's beautiful. And let's see what it's like down in north central Florida, the home of Richard Condit.
It's beautiful here too. It's a few clouds, but it's sunny and warm. It's very nice.
Welcome back. Hope you're okay.
I'm just fine, and it's great to be here.
Good. We missed you. And then finally, from another place on the East Coast, our guest today from Philadelphia is Michael Bouchard. Welcome to TWiV, Michael.
Thank you.
How's the weather in Philly?
It's beautiful.
Great. Who's coughing?
That is my wife in the background. Sorry about that.
Would she like to join the TWiV today?
No, I probably not. I only have one headset, so.
Today, we are going to talk about Michael's field, which is Hepatitis B virus. This was prompted by a Drexel student. So I should say that Michael is an assistant professor. What's the department, Michael?
The Department of Biochemistry and Molecular Biology.
Biochemistry, it's right in front of me. Molecular biology. Drexel University College of Medicine. And his laboratory is interested in livers, and in particular Hep B virus, which infects the liver. We got an email some time ago from Allison, who's a student at Drexel, and she wrote, I'm currently taking a special topics course in virology, and this week's lecturer mentioned previously working with polio. When I looked him up, apparently he's a former student of yours, Michael Bouchard. Just wanted to share. Why don't you invite him on TWiV? I don't recall you doing an HBV episode, only HCV.
Oh, that's why his name sounded familiar.
You guys must know each other, right?
Yes.
Small overlap.
Small?
Yes, small overlap.
Yeah, so
just
define small. I want to know what's small.
We sat at lab benches directly across from each other for I prefer not to disclose how many years while we were both in graduate school.
Multiple years.
Yes.
Okay.
Yeah, this is a little insider virology
It's a cozy
today.
little group, yeah.
Yes.
Yeah,
yes.
but that's okay because we just look for people working on interesting viruses. And
Michael and I had a good arrangement because we both like to keep our benches neat. So
Me too.
Yeah.
Wow.
all right. Well, me too. We got all neat freaks today. All right. Michael, you still working at the bench?
I am not. I have not worked at the bench in about 3 years.
Wow.
No time.
Do you miss it?
I do. It's a struggle with having to write grants all the time and teach when what you really want to be doing is really back in the lab working at the bench. There's just no time to do that anymore.
Yeah. Well,
It is difficult.
when you get old, you can do it again. Both Rich and I, occasionally tinker.
Well, the students don't want me coming in the lab anyway. They tell me I screw everything up.
That's right.
All right.
sounds
They're
like exactly the relationship we had with Vince.
Exactly.
Yeah, stay out of the lab. Well, I should say that after you got your PhD in my lab, you went to do a postdoc with Robert Schneider at NYU, and he is where you learned about hepatitis B virus, correct?
Correct.
And perhaps we'll get to talking about what you did there and what you're doing now. But I wanted to start talking about Hep B because we haven't talked about it on the show. Let's introduce it. Maybe we could start with where it came from. How was it discovered?
So a little bit of the history. So I think that before you start talking about the virus, you sort of have to talk about two things. One is you have to define hepatitis, which is an inflammation of the liver. And the other is you have to think of the clinical consequences of that, which can be, for example, fatigue, malaise, but also jaundice or yellowing of the skin. And it's important to think in that way because that's the early descriptions of hepatitis were really clinical descriptions. And so if you look through sort of the historical context of hepatitis, you see that for probably for thousands of years. It was known that there were these disease-causing, or there was a disease associated with this yellowing of the skin. And that tended to be apparent in areas with poor sanitation. It tended to travel with armies, so it was a disease of warfare. But I think from what I can find, the real description of Hep B, or the clinical disease that's probably associated with this virus, was really in the late 1800s, and it was the recognition of a hepatitis that was transmitted through, in that case, a smallpox vaccine, and its incubation period, or the period between when the infection occurred and when the actual symptoms were apparent, was much longer than classically thought for the acute hepatitis, such as hepatitis A, which is one of the other major causes of hepatitis. And so the incubation period was about 2 to 8 months. And so it sort of initially, so it set it up as this different kind of hepatitis. And then following that, there were some outbreaks again of vaccine-associated hepatitises. There was a huge outbreak in the U.S. Army in the late 30s that was associated with a yellow fever vaccination. And for many of these vaccines, they used actually human serum as a way of stabilizing the vaccine. So it became associated with something basically that was present in serum. And the discovery of the virus itself is actually kind of interesting, because it was discovered, one would say, almost accidentally. I mean, Baruch Blumberg had designed a series of experiments in the early 60s where he was looking for essentially inherited polymorphic traits in different geographic areas. And the hypothesis was that if you took blood from multiply transfused individuals and used it essentially to do what we would now call a Western blot on blood samples from different parts of the world, the idea was that the multiply transfused individual would have probably antibodies to proteins that he or she had been infused with that were different. And in this screening process, he identified an antigen that reacted to serum from an American patient, essentially, and the serum was from an Australian Aborigine. And so he referred to it as the Australia antigen.
Aha. I've heard the name before. Now it makes sense. Great.
And so he began to characterize where was this Australia antigen, what other parts of the world, and he found there was a high presence of it in some African and Asian countries. About that time, Alfred Prince's group was also studying hepatitis B and was using serum from hepatitis B positive patients to essentially do immunohistochemistry on liver samples. And essentially, the end of a very long story is that the two fields kind of came together and realized that they were both looking at the same thing. What also helped in this process, which is not a great story, is that someone in Baruch Blumberg's lab, who was Australia antigen negative, became Australia antigen positive after experiencing a bout of hepatitis. So they kind of put the whole story together, and it became recognized that the Australia antigen actually was one of the envelope proteins of the hepatitis B virus, which is secreted at very high levels in the serum of individuals who are chronically infected with the virus. And that really allowed for, first of all, it set up a way to screen the blood supplies, which was obviously very important for preventing future contamination of the blood supply with Hep B, but also allowed a lot of retrospective studies that really confirmed a hypothesis that had been around for quite a long time, which was that this new kind of hepatitis, which was now called hepatitis B, was actually associated with the development of liver cancer. And when they overlapped basically the map of what parts of the world had the highest prevalences of liver cancer and what parts of the world had the highest prevalence of this Australia antigen, or this S antigen, which is what it's called now, they essentially overlapped, showing a very high correlation with areas of the world that had Hep B infections and area of the world that had high rates of liver cancer.
So, Michael, when Blumberg started working on it, was it already called Hep B?
It was, and it had been called Hep B a little bit earlier, actually in the mid 40s, I believe, and it was really to separate the idea that there was a hepatitis that you were infected and you showed symptoms very quickly, so very acute hepatitis, and then a hepatitis which had a longer period of incubation before the symptoms came up. But I think in that early, even though it was referred to as Hep B, you couldn't really say that it was specifically the virus we call Hep B now, because there are other, for example, Hep C, causes chronic infections as well and a longer incubation period.
And of course, Blumberg got the Nobel Prize for that discovery, right?
He did. Yes, he got the Nobel Prize in medicine for essentially discovering Australianogen, showing that it was Hep B surface antigen, and then really providing a means for screening the blood supply. It also provided a means for developing a vaccine, which was originally developed by he and some other researchers by using serum from chronically infected individuals and basically purifying S antigen from that.
Okay. So what does the virus look like?
So the virus is an envelope virus. Within the envelope is an icosahedral capsid, and within the capsid is the virus's genome. The genome is a partially double-stranded circular DNA genome. So it's replicated by reverse transcription, and one of the strands is complete, the negative strand, and the positive strand is completed to varying lengths. So we refer to it as partially double-stranded.
Is there a reason for that you can think of?
Yeah, so there are some ideas. I mean, it's one of those, it's a hypothesis that's out there. I don't think anyone has definitively proven it, but the idea is that as the virus is replicating, so it replicates entirely in the cytosol, entirely in those capsid structures. And it's thought that basically it's taking nucleotides, deoxynucleotides from the cytosol as it's replicating, and as part of its replication process, it buds into the ER. And one thought is simply it runs out of nucleotides as it's budding in and acquiring its envelope. So it basically keeps completing the process until it runs out of nucleotides. We know that if we take those purified capsids and actually just give it nucleotides, it will in fact finish the second strand.
Okay, so this touches on another question. I may be jumping ahead of you, but I feel compelled to ask this now. The envelope is acquired intracellularly, correct?
Yes.
Not by budding.
What we say is that the virus buds into the ER. So the envelope proteins are actually embedded in the membrane of the ER. And as the virus is replicating encapsulated within this core, it essentially moves to the ER and interacts with the envelope proteins and buds into the ER and then is secreted from the cell.
Okay, so basically the idea then is the ER is continuous with the plasma membrane, so I can make my way out from the inside of the ER. Is that the idea?
Well, so we used to say that the virus was secreted by the constitutive secretory pathway, but there is now evidence that it uses the vesicular transport pathway. So I would say no, it's probably not that the ER membrane is contiguous with the plasma membrane.
Okay, so that you move on from there to a vesicular pathway to be secreted. Okay, fine.
Right.
Great.
I have to interject. My textbook, there's a picture of HBV budding into the ER, and it's got an error. It has the glycoproteins on the cytosolic side of the ER. All right, we never picked it up. So I took that picture and put it in my final exam for my virology course as an extra credit.
What's wrong with this picture?
Right.
That's good.
Bad, right? Well, it's extra credit anyway. Michael, why does it have a reverse transcriptase? It does not integrate like retroviruses do, correct?
Right. So why does it have a reverse transcriptase? I guess that's a good and open question, and I don't know that anyone knows why it replicates through reverse transcription versus some other mechanism. But the answer to your question is yes, it does not integrate. In fact, integration is not a requisite step in replication. In fact, when the virus brings its genome into the nucleus, the partially double-stranded circular DNA genome is repaired in the nucleus to a covalently closed circular DNA, essentially a plasmid or an episome in the nucleus. And that now serves as the template for transcription. Those transcripts are exported to the cytosol, and then one of those transcripts in the cytosol is encapsulated and then converted into DNA in that capsid in the cytosol.
Right.
There, you can get integration. And in fact, when you look at many HPV-associated tumors, you can find integrated HPV. But that's really considered to be an aberrant step. It's not required for replication.
I have to say, when I first learned about hepatitis B replication as an undergrad in a microbiology course, I thought this was the most bizarre thing that I could imagine. It's got its DNA genome sitting there in the nucleus where all the replication machinery for the cell is, but it makes an RNA copy of it and sends it out to the cytosol to be reverse transcribed.
Yeah, it kind of thumbs its nose at all the nuclear stuff.
Right.
Right. In fact, the pool of cccDNA in the nucleus does not come from replicating the cccDNA. It actually, in the beginning, a certain percentage of the encapsulated RNA that's being converted to DNA gets redirected back to the nucleus to build up that pool. And then it's thought that, at some point as the pool of core protein increases and envelope protein, et cetera, it starts diverting it down the pathway to get secreted from the cell.
So do you think this was a retrovirus as we would think of it, maybe integrating the genome at some point in the evolutionary past, or?
Yes, if you look at some of the earliest papers, and I'm forgetting the name of the author, but one of the earliest papers where they really looked at the structure of HPV, they kind of referred to it as a defective retrovirus. And when I teach HPV, I purposely will put it linearized. I will linearize the genome for the students underneath a retroviral genome, and it actually looks quite similar. It has basically, the structural protein for the capsid, the envelope protein, and the polymerase protein, and then an accessory protein in the case of the mammalian hepatinoviruses, which is the X protein. You can line it up almost exactly with almost a shortened version of a retrovirus. And so some of the early papers suggest that it's, they called it a defective retrovirus. I'm not sure that most people in the field would refer to it as a defective retrovirus anymore, but that was one of the original statements.
It's insulting almost, right?
Yeah, it seems to get by just fine.
Yes, yeah.
So, Michael, when you said that the gap DNA gets in the nucleus, it's repaired. Is that done by cell enzymes?
So you can actually inhibit the activity of the viral polymerase, and you will still repair that partially double-stranded DNA. And the repair involves more than just extending the strand. You actually, as part of its replication cycle, it actually has a protein attached to the 5' end of the negative strand of DNA. So that has to be removed. The pregenomic RNA, which is what is reverse transcribed, is actually terminally redundant, and the terminal redundancy also has to be fixed to some extent. So it's more than just filling in the gap, and you can shut off the polymerase of the virus itself, and you still get repair of that. The process of repair is not all that well understood.
So you mentioned the vaccine, are there also antivirals for Hep B?
There are. They're basically nucleoside analogs.
They target the RT, I presume.
Exactly. And so you can treat an individual obviously with interferon, which was some of the earlier, and I think that it's still used, but of course there's a lot of side effects with that. So there are nucleoside analogs, but the virus, because it replicates through reverse transcription, has a fairly high rate of developing resistance. So even some of the more recent drugs where the claim is that the level of resistance or the time to mutations that will cause resistance takes longer, still eventually, I think we could predict that eventually you'll develop resistance to those nucleoside analogs.
So is the RT very similar to a retrovirus reverse transcriptase, and do the same drugs work on both?
That's a good question. So in terms of whether it's similar, yes. It has the polymerase domain, the RNase domain. It has a difference in that at its N-terminus, it has what's referred to as a terminal protein. And that really, so unlike retroviruses, which use tRNAs as primers in their replication, HBV doesn't need a primer. In the sense that it doesn't bring in any short nucleotide sequence. It actually, the terminal protein interacts with a structure in the RNA and essentially kind of copies off the strand of RNA. So the protein is actually covalently linked to the 5' end. So that is one difference. But in terms of the polymerase domain and the RNase domain, the RNase H domain, it's very similar to retroviruses.
Okay.
And you asked me about the drugs, whether they, I don't know the answer to that. I would think that they would, but I'm not sure.
Okay.
So roughly when was the vaccine introduced?
I think that Bloomberg's was, I think it was the 70s or 80s. But then there was concern with that vaccine because it was derived from essentially the serum of chronically infected individuals. So they then generated a recombinant vaccine. It turned out that all you really need is the S antigen, so this envelope protein. So they cloned a DNA copy of the S antigen into a yeast expression system, and you essentially grew up tons of this protein. And the current vaccine is actually really it was the first recombinant vaccine where they essentially expressed a single viral protein and purified that and used it as the vaccine.
One could also. Who was the lead scientist who did that?
I do not know.
Sorry, it's just somebody we've talked about a lot on TWiV.
It is, and who was that?
Yes. Maurice Hilleman.
Oh, I didn't know that.
Yes. Oh. Yes, another Philadelphian.
So what did that do to global Hep B burden?
So in where it's been instituted as part of comprehensive vaccination, it has really dramatically decreased the incidence of the development of chronic infections or infections with Hep B. It's also had a dramatic effect in those areas on the level of liver cancer. So I think the real good example of that is in Taiwan, where basically everyone now is vaccinated for Hep B, and the rates of chronic infection have dramatically decreased, as have the rates of liver cancer. The issue, though, is it's an expensive vaccine, and it requires 3 shots. So you get one at the first, 1 times 0, 1 at 1 month, and 1 at 6 months typically is the schedule. And when you're trying to bring those kinds of vaccinations to, for example, third world countries, it can be very difficult based on the stability of the vaccine as well as follow-up. So there's still a significant level of chronically infected individuals in the world and still a high rate of infection.
I'm looking at some numbers here. It says that currently a third of the world's population have been infected, 2 billion people, and there are 350 million carriers.
Right.
So so there's not everyone who's infected becomes a carrier.
No. So it depends on your age. So chronicity is really so basically it's really defined as the long-term expression of the S antigen. So you get continual viral replication, but clinically you detect that as a long-term expression of S antigen. Typically, there are always these deviations from these rules. But in adults, if you're infected as an adult, which is predominantly, so the virus is spread bloodborne, body fluids, sexual transmission. Adults, Western societies, one could say predominantly, although I'm sure someone would disagree with me, a lot of sexual transmission. Only about 5% of adults will go on to develop a chronic infection, and it's thought that a strong immune response prevents chronic infection. If you're an infant born to a chronically infected mother, it's not typically thought that infection occurs through the placenta, for example, but rather through blood exposure as the baby is being born. Infants who are exposed to Hep B have as high as a 90 to 95% chronic infection rate. And it's really those people that are the largest group of carriers. So an enormous problem in Asian countries where there's a significant level of chronic infection. They've done a lot now to treat chronically infected mothers, really to lower the viral load, to prevent babies from becoming infected. But prior to those policies being put into place, really the major source or the major population that was chronically infected were those infants that were born to chronically infected mothers. And they're really the ones that often go on 20, 30 years later to develop liver cancer.
And is that the reason for starting the vaccination schedule at day 0?
Right, exactly. It's the reason now for, immunizing pretty much immediately. Yes.
Right, because that chronic infection is so high in those individuals.
Yes.
So if you're chronically infected, you may or may not have episodes of disease down the road. Is that correct?
That is correct. You can go through periods where you have absolutely no symptoms whatsoever. Basically, the symptoms are really associated with the immune response.
Okay.
The immune system basically destroying infected cells. So the virus itself is not cytopathic. It's really the immune system trying to clear the virus, and obviously it's destroying liver cells and all the consequences of destroying liver cells. So the disease really is a response to the immune system really destroying the liver cells. And you can definitely go through periods where you're not getting a strong response, maybe the virus isn't replicating as high, and then periods where you get this reactivation and this strong immune response where you now show those clinical symptoms of hepatitis.
So this, to me, is the most insidious type of infection, where you have somebody who's infected, shedding infectious virus over a long period of time, but asymptomatic.
Yeah, it's actually a little more. The statistics actually, Vincent, are a bit worse than what you were just saying, in the sense that so the estimate is that yes, about a third of the world's population has been infected, and that about 350 to 400 million people remain chronically infected by our current definition, which is really the ability to detect the S antigen. But there's now a recognition that many individuals who were thought to have cleared the virus, in fact, have not. It's just that the level of virus is below what one might consider the standard techniques for detection, so that you can't detect by the standard techniques the presence of S antigen. But if you do very sensitive PCR studies, you can actually show that quite a few individuals retain the virus in what's referred to as an occult infection. So the virus is there just at very low levels. And there's now a recognition that those individuals as well may have an increased risk for developing liver cancer. And there's also recognition that sometimes that's the cause of problems when one transfects a liver. So they'll take a liver from someone who is anti-core for HBV, so anti-HBV core positive, which says that they were previously infected with the virus, but you can't detect the virus by the standard techniques, and then they transfer the liver into someone who needs a liver transplant, and of course they shut down the immune system so you don't get rejection, and up pops the virus. So that it was there all along, just below the level of detection.
Wow. Is the liver cell the only cell that the virus replicates in?
It is the predominant cell. There are some individuals who claim that there are some, I believe it was polymorphonuclear cells, but some blood cells that can be infected. But there's a lot of debate in the field as to whether that represents a true infection or sort of sticking of the virus to the cells or not. A non-specific uptake of the cells.
Right.
And is it
of the virus.
Is it known what dictates the preference for liver?
So it's predominantly at the level of receptor expression. That's what's currently thought. And I say that with a little bit of hesitation because we don't know what the receptor is. But the thought is that these viruses in this family have very narrow host range. And it's believed that the host range is predominantly defined by the receptor, as well as the cell type that's infected. But there's also evidence that factors within the cell, transcription factors, for example, also regulate the cell-specific expression of the viral transcripts and proteins.
So are humans the only host in nature of Hep B?
Humans are the only natural host of the human hepatitis B virus. Yes, you can experimentally infect chimpanzees and some other non-human primates, but naturally it is humans. But there are many other animal models for this virus family.
There are hepatitis B viruses of other animals, right?
Right.
Ducks and so forth.
Ducks.
Right, exactly. So there's the AV hepanoviruses, which are those that infect birds, and then there are the ortho hepanoviruses, which are those that infect mammals. One of the very important animal models is duck. Another very important animal model is woodchucks. A significant number of woodchucks in North America are chronically infected with the woodchuck hepatitis virus, which was actually originally discovered here in the Philadelphia Zoo. Some of the woodchucks were dying, and when they did autopsies, they discovered that they had liver cancer that was very similar to liver cancer in humans infected with HPV. They eventually isolated the virus. The virus for woodchucks is really devastating. 100% of chronically infected woodchucks will develop liver cancer within about 2 or 3 years of their infection.
Can you grow this in culture?
You can. Well, you can. Yes and no. So you cannot. There are no available cell lines. Let me buffer that a little bit. There was a report recently of one cell line that you can do a direct infection on, but that infection is extremely inefficient. So I think we can still safely say there's really no good tissue culture system for directly studying a direct infection with HBV. But you can bypass the infection by transfecting the viral genome directly in cells, and you can get a complete cycle of replication within the cells. You can directly infect for a limited time cultured primary hepatocytes derived from the host that is specific for that particular member of the hepanovirus family.
Okay.
Now, you're also working on some culture systems that might be useful for this, right?
We are, yeah. I mean, we use in my lab we use cultured primary rat hepatocytes. For that, we have to bypass by directly transfecting the genome in, either by transfecting naked DNA in, or the typical transfection reaction with liposomes, et cetera, or we make recombinant adenoviruses. So we've inserted a DNA copy of the viral genome into an adenovirus. Adenovirus is extremely good at infecting liver cells. So the adenovirus is replication defective, but carries the genome of HBV. We also have some tissue engineering projects that are going on where we're engineering in microfluidic microchannels human hepatocytes with a long-term goal of being able to do direct infections with the human virus.
There was a company that I just interviewed for an article I did last month that is working on something kind of similar. They're nano-engineering surfaces to grow hepatocyte spheroids. So it sounds like maybe you're doing something kind of similar.
We're not using nanotechnology for what we're doing, but we are. It's sort of a similar idea in the sense that we're trying to recreate the microarchitecture and microenvironments of the most basic unit of a liver, with the idea that that would facilitate long-term survival of differentiated human hepatocytes so that we could actually do a direct infection and study the infection process.
Right. Kind of makes you miss polio plaque assays, doesn't it?
It certainly does.
No plaques. No plaques. What are you going to do without plaques?
Not only no plaques, you can't do an infection.
Oh.
Right.
So can these animal hepatitis viruses infect people?
No. So again, very narrow host range. So even though the viruses are very similar, they do not jump species.
So Hep B is an eradicable disease then, right?
Yes, you could. If you could vaccinate. Well, yes, in long-term theory, yes, it could be. What we are discovering now, though, is there's a certain percentage of people that don't respond to the vaccine. And there's also the recognition that we're beginning to see the emergence of vaccine-resistant mutants of the virus. So yes, you could eradicate it, but I think it would probably require more than just the current vaccine.
Right. I have two big wonders here, and one of them is, how does the virus persist in the face of, our immune response?
So I think that our understanding is that, recall that that hepatocytes have a very long half-life. So the average lifespan of a hepatocyte within your liver is about 6 months. And if you would estimate that each of those has, 20 to 50 copies of the CCC DNA, that the immune system is trying to attack that, but the liver remembers a regenerative organ, so it's trying to regenerate. Basically, the thought is that those CCC DNAs are extremely stable and very, very tough to get rid of. In fact, that's part of the issue with long-term treatments with the nucleoside analogs, because you may totally get rid of viral replication, at least that you can detect, but the CCC DNA is still sitting there. And it takes so long for the liver to turn over and to get rid of all that CCC DNA that it may require extremely long treatments, and by then the virus has popped up as being resistant now to the treatment.
So
And
even though it's
at the
not
same-
integrated, it presents a problem similar to what you see with HIV.
Yes, in the sense that because the hepatocytes are so long-lived, and there are multiple copies of that CCC DNA, it's extremely hard to get rid of.
And you've also found, I guess in your most recent paper, it looks like the virus is also maybe stimulating the cells to reproduce.
Well, what we think it does is that the environment of a hepatocyte is probably so basically hepatocytes differentiate and then they become quiescent. They sit in G0 and they perform their functions. And one idea is that this is a DNA virus. It needs to replicate its genome. That environment is probably not all that favorable for replication. So it's going to move the cell, like many DNA viruses, out of quiescence into the cell cycle. And so what we think the virus does is it bumps cells out of quiescence and yet holds them in G1. So it creates an environment that facilitates its replication without actually pushing the cell all the way into the cell cycle, which we think is because it's a pretty clever virus and it doesn't want to compete with the cell for the machinery that it needs to replicate, such as nucleotides.
Right.
But there is as far as we know, there's no specific immune evasion mechanism.
Not that there's been a search for that, and that's a lot of Francis Cesari's work. I don't think the virus per se has an immune evasion, but clearly the immune system plays, and that's a lot of his work, plays a critical role, both in the development of a chronic infection, and in this case, the absence of a strong immune response, as well as the long-term progression to liver cancer. It's really thought that the inflammatory response is a key contributor to the development of HPV-associated liver cancer.
So how does the virus cause cancer?
Good question. If we knew that, I wouldn't have any more research.
I didn't figure I was going to get a quick answer to this.
So obviously there are lots of theories. Clearly, as I just said, I think there's pretty universal acceptance in the field that the inflammatory response plays a key role. So we have, the liver is a regenerative organ, and we have an immune system attacking HPV-infected hepatocytes. We have it trying to destroy those hepatocytes, and we have the liver turning over. And one idea is that in the context of an inflammatory response and all of the associated consequences of an inflammatory response over time, we can select for transformed cells. We don't think that that's the only thing, though. So in this family of hepatoviruses, There are those that infect birds, as I said, those that infect mammals. The bird viruses all cause chronic infections as well, but none of them cause cancer in their respective hosts, whereas the mammalian viruses, which also cause chronic infections, can cause liver cancer. And one of the key differences between the AV hepatoviruses and the ortho hepatoviruses is the presence of basically we call it an extra protein, the X protein. So named because when it was discovered, no one knew what it did. It didn't have any homology to any other known protein. And there's a lot of evidence that it also can contribute to the development of liver cancer. So if you make a transgenic mouse that expresses the X protein, you either get, depending on the strain of mice, which tells you more that it's probably a cofactor, you can get the direct development of liver tumors, or you can get an increased sensitivity to carcinogens. We also know that if you knock out this protein, at least in the case of the woodchuck virus and the human virus, they don't replicate. So it's thought to play a key role. It activates a lot of signal transduction pathways. It can modulate apoptosis, cell cycle. So it's thought that it contributes to this process, and it's possibly the combination of what it's doing to cells, as well as in the context of this continued inflammatory response, that you eventually begin to select for transformed hepatocytes and progress to the development of liver cancer.
Is there a causal relationship between viral DNA integration and cancer?
So in the woodchuck model, there's very good evidence that integration is probably linked to the development of cancer in the woodchuck model. So I think the current estimate is as high as 50% of woodchuck liver tumors associated with WHV infections have an integration in the vicinity of the NMYC2 gene, which basically, if you deregulate MYC, you're going to deregulate cell cycle progression and apoptotic pathways. So it's thought in the case of the woodchuck that can have a contribution, but then you've got the other percentage that don't have integration there. In the case of the human virus, there's no indication of a consistent site of integration. So it's a hard question to answer. I mean, it could integrate and be an initiating event, but because there's no consistent site, it's hard to prove that. However, when you look at tumors, you often see that the tumor is monoclonal. So you have the same integration site in the entire tumor, which has been used as an argument to suggest that it may have some role in one of the early events. But again, it's very tough to prove that.
So I'm thinking about a situation like in the human papillomaviruses and cervical carcinoma, where it's thought that the integration both deregulates the viral oncogenes and installs them permanently in the cell containing the integration. Is there any thinking that something similar could happen with hepatitis B virus?
Yeah, so you're referring to E6 and E7. So
right.
for HBV, again, there's quite a bit of debate about this. So a significant number of tumors that are associated with HBV infections, you can find that there is integration of the X gene. And so that's been used as an argument to suggest that, that X is being expressed and then is having an effect on the process of transformation. I think that, again, that's very hard to prove because in many instances where they've shown integration, they haven't actually demonstrated that the protein is at all expressed. So basically you get the integration of the sequence, but you have no idea whether you're actually even expressing the protein. So tough to prove that. But there are definitely those out there that argue that it may be contributing to the process.
So if you put just X gene in a cell by itself, does it transform the cell?
It can, depending on the cell type. And so in some, you get variation from cell type to cell type. What confuses the issue is that if you do, in many cell types, if you do something like a colony formation assay, you actually get X inhibiting it. which in those cases, it's been suggested that's really because X is causing apoptosis. In the context of the liver, that actually might in itself contribute to the development of liver cancer because you're getting induction of apoptosis and the liver is going to regenerate. So you may over time then select for cells that are resistant to those apoptotic signals.
So there's no clear mechanism for X, it sounds.
No, and you can, so, for example, X activates NF-kappa B, and you can argue there, if you look in the literature, there's arguments for elevated NF-kappa B in the context of liver cancer. But there's also papers that show that if you inactivate NF-kappa B, you get liver cancer. Again, with the idea that the liver is going to regenerate. So it's going to find a way to get around that inactivation. And what we see is that in primary cells, X activates NF-kappa B to inhibit apoptotic pathways. But if we block NF-kappa B activation, we actually get induction of apoptosis. That's mediated by X. And you could argue for the development of cancer down either route.
For other tumor antigens like the polyomas, even the oncogenes of retroviruses, so they have cell cycle promoting effects, right?
Right.
And those proteins are present in the transformed cells. They either activate a promoter or the protein itself is present. So in all people with liver cancer, is the X DNA there, and is the protein made?
So again, you would hear a debate on this if you asked a number of scientists. I would say no, that it is not present in all tumors of HPV-associated liver cancer. And in fact, we might consider that it's more of a hit-and-run mechanism, where potentially it initiates a series of events that contribute to the process, but doesn't need to stick around once that has started.
Okay. So you're working on X still, right? Is that correct?
So yes, a significant part of the lab works on trying to understand what the role of X is, both in HPV replication as well as in the development of liver cancer. First question is a little bit easier to answer because we can directly assay replication. The second is a little more difficult because it's very difficult to develop assays, especially cell culture assays in primary cells to look at transformation.
So of the virus, well, you can't infect cells, as you said. You have to put DNA in, right?
Right.
But if you put DNA in a certain cell type, would it transform it?
Again, there
Sometimes.
is some. Yeah, exactly. There are definitely some studies that show that you can get that to occur.
Yeah.
But again, it's very cell type specific. It also seems to require other, for example, you may need to have T antigen around or some other activating oncogene sitting there as well. X on its own, I think one would be hard pressed to argue is an oncogene.
Yeah. But maybe together with some other aspect of what the virus does. I mean, the chronic inflammation, as you said, plays a big role, right?
Right,
Yeah.
exactly.
So you mentioned T antigen. Is there any evidence that another infection also contributes?
The only types of co-infections that I think people have focused on are HBV and HCV. And the studies that I've seen sort of suggest that one doesn't like the other one to be around, so that HBV seems to downregulate HCV and vice versa. What's interesting is that for viruses that are so different, they have proteins that do very similar things. So the HCV core protein, you can almost line up many of its functions alongside X. X elevates calcium in cells, so does core. X regulates reactive oxygen species, so does core. The consequence of that is an impact on cell cycle and apoptosis for both proteins. So it's as if the viruses are doing very similar things in the cells. So when you have both together, obviously there is a problem. The other virus that causes a significant problem is the hepatitis delta virus. So HDV. And HDV requires HBV to be there in order to replicate. In the absence of HBV, you cannot get any HDV replication. And people who are chronically infected with both HDV and HBV have a far worse disease and a higher progression to liver cancer.
And their disease is really an acute disease. It's like a real hepatitis, right? If you're co-infected with both.
I think I don't know if I think the problem is really the long-term effect in terms of progression to liver cancer. I know that the clinical symptoms are far worse. Whether one would classify that as consistent with an acute infection or not, I'm not sure.
I see.
Delta is almost like a parasite of a virus, right?
Right. It basically needs It's not going to do anything if HBV is not around. Which when you think about it, that means you kill two birds with one stone. Essentially, if you eradicate HBV, you get rid of HDV as well. So vaccination against HBV also is like being vaccinated against HDV.
Are there hepatitis delta viruses of other animals?
Plants.
Well, yeah, they're viroids, right?
No, they're satellites. Delta is a satellite, which is different from a viroid.
Okay.
And there are satellites of plants. Delta is the only animal satellite, as far as I know.
Yeah, I don't know of any other.
Okay.
Yeah.
I just taught this, Rich. That's the only reason I know.
Teaching helps.
Viroids, satellites, and then the other odd one is the last lecture of the course, prions, right?
Okay.
But there are 18 million people infected with Delta globally. Yeah.
Yes, and they have a far worse disease than just B alone. It's pretty devastating.
So that's actually a fairly small percentage of the total number of people who are infected with hepatitis B.
Yes.
Okay.
Yes.
Because Delta can't get by on its own, presumably the only way to get it is to get it in combination with Hep B. So you'd have to get it from somebody who's co-infected.
Or, yeah, that's a good point. You're right. Yes. They would have to have
Sometimes
both.
I have them.
Yes. I hadn't thought about it that way, but I was thinking you were going to say you have to have one, you have to have B before you get D, but actually you're right. You get them both at the same time or D after B, but yes, you've got to get it from, obviously someone had to have had both.
Right.
Of course, we should mention that in cell culture, if you take Delta and put it in cells, it will replicate. It will make RNA. It won't encapsulate without Hep B.
Right.
Because the RNA uses the cell polymerase to replicate. It's a great beast.
But the Delta does make something that is of value to it, right?
Yeah, it makes
Delta antigen.
Yeah, that's
and
right.
what does that thing do?
You asking me?
Do you know,
Anybody.
Michael?
I don't know, actually.
Delta has two roles. One is it participates in viral RNA synthesis. There are actually two forms of Delta made, and one has a role in RNA synthesis, and the other helps it to be encapsulated into the Hep capsid.
So when you say it helps in RNA synthesis, does it somehow help the cellular RNA polymerase work on this RNA genome?
Well, the first round, it's not there, right? So it can't be essential, but it's thought to help it later on be more efficient. Yeah.
Right.
And then the other version helps to encapsulate the genome. Delta also coats the genome. So the one Delta coats the RNA, then the second Delta links it to the Hep B capsid. Well, that's the idea anyway. We'll have to have John Taylor on.
I was just thinking that's the perfect person to ask all those questions to.
He's down there in Philly.
He's at Fox Chase.
Fox Chase, right. And he's one of the Delta persons in the world. It's really interesting. Great. Anything else that we can ask Michael? We could move on to email if not.
I'm curious as to whether you can tell me whether the vaccine has had a significant effect on HBV in the United States.
So actually, the rates of HBV. So the last statistics that I looked at is that the rates of HBV in the U.S. Have actually started to rise, but it's thought that that's not domestic, but actually immigration.
I see.
And immigration from countries with chronic infection.
Okay.
So I think overall the rates in the U.S. were not high anyway.
Okay.
And obviously now it's part of our vaccination strategy that everyone is vaccinated against HBV, except for our age group is the one I think they've missed. But now, to get into college, get into high school, get into elementary school, you have to be vaccinated. Every place where it's been instituted that had high rates of chronic infection, the rates have gone down quite
Okay.
dramatically. Again, I referred to earlier, I think the real prime example of that is Taiwan, which had an enormously high rate of chronic infections, where a lot of the early studies were done to really demonstrate the link between HPV infections and HCC. And universal vaccination there has really dramatically decreased the rates of, or the amounts of chronic HPV infected individuals, as well as the levels of HCC. In the US, again, as I said, the last statistics that I looked at suggested that liver cancer itself is one of the few cancers in the US that still is on the rise. And that the incidence of HBV itself hadn't declined all that much, but that was really due more to immigrants than to the domestic population.
And this presumably is something where it would take decades to see the chronic cases disappear, right?
Right, because the vaccine doesn't do anything if you're chronically infected. At least, there have been attempts to use vaccine strategies to deal with chronic infections, but to my knowledge, they haven't been that effective. So if you're chronically infected, the vaccine's not going to really do much for you.
So we'd probably expect to see in Thailand, where there was a high rate initially, they brought that down dramatically with the vaccination, and then we'd expect to see a plateau that's going to go on for a long time.
Right. And I think it'll take years to also really see such a huge effect on the rates of liver cancer as well.
Is there any effort to develop therapies that would help chronically infected people, like RNAi or something like that?
Yes, there are. That's actually a major drive in the field. Obviously the nucleoside analogs are one, but we also believe that X is a prime target. If you knock down the activity of X, the virus can't replicate. So you could, in essence, we look at it as, could we create a cocktail in the same way they have for HIV, where you targeted multiple viral proteins in individuals who were chronically infected. So there is definitely an interest to try to treat people who are chronically infected. The current problem is that with the limited number of limited therapies that are available, the virus just develops resistance very quickly.
And for targeting those therapies, X probably does mark the spot.
Yes.
You've been awfully quiet there on that front. That's very good.
Well, I let woodchucks and ducks pass without comment.
You're always welcome. People love it.
Yeah,
Anyway.
Some do, some don't.
That's true also. That's a great virus. I like it. Well, I never found a virus I didn't like, to be honest. Sorry, Michael, what were you going to say?
I was going to say it's a clever virus.
Is it good for funding?
It's interesting. It's probably the same problem I recall having when I was a PhD in a polio lab, where I always got the argument, but there's a vaccine. Why do you need to study this anymore? And I constantly remind people, first of all, that the enormous number of chronically infected individuals, but also the idea that it's an incredible tool for studying processes involved in the development of liver cancer. The estimate right now is that 80% of liver cancers are associated with viral infections, and the vast majority of those are associated with HPV infections. So it's a powerful tool for understanding, what has to happen to a hepatocyte to move it down the transformation pathway. But your question was funding, and you asked me that question in about 2 months when I hear back from the response to my grant application.
It's interesting that you say that most of the cancers are due to Hep B. I'm on the study section, and I see very few Hep B grants and huge numbers of Hep C grants.
Right.
Now, that may be because you can now grow Hep C in culture, so everyone's jumping on that.
I hope that I think I would hope that might be a reason, but I also think that research sometimes goes where the dollars appear. And HCV is, in this country, obviously a problem that's much of a higher magnitude than HBV is. HBV really is an enormous problem in Asian countries, in Africa. China, 10% of the population is chronically infected. Parts of Africa, 20% of the population is chronically infected. But in Western countries, the rates or the number of chronically infected individuals is quite low. So it may be that it's also not viewed as much of a problem because of that. On a worldwide level, it's far more significant. The rates are double in terms of chronic infections of HCV, and the progression to liver cancer is much, much higher than HCV.
Okay, I would like to do a few emails, but before we do that, I need to acknowledge the support of Data Robotics, the makers of Drobo. And we've talked about Drobo many times. It's a wonderful storage solution for your computer. It consists of basically drive arrays. They're little boxes where you can slip 4, 5, or 8 hard drives into, and your data are duplicated across the drives and protected so that if any single drive fails, you can simply take out the dead drive, put in a new one, and your data are still present and they're copied to the new drive. And we use Drobos here. I use them in the lab. I use them at home. They're terrific solutions. They make many different models, and I thought I would talk about the Drobo today. The Drobo is the original model they made, the 4-bay model. It's pretty much the expandable storage for everyone. You can put your iTunes library on it, your photos, videos. If you have a lot of large photographs or video footage, it's a perfect solution for that. And 1/3 of the 4-drive Drobos that are in use are used by corporations to back up their servers. So they're really handy in a number of environments. I have one right here in my office that I use to back up all of our critical data. And they start at $3.99 without drives. And of course, you can get some money off of Drobos. If you go to drobo.com/TWiVpod, you will see you can get $100 to $750 off of a Drobo unit. And we are also giving away a 3rd unit on TWiV, and we'll soon announce that contest. So drobo.com/TWiVpod. We thank Data Robotics for their support of This Week in Virology. I thought we'd do a couple of emails now. We have a bit of time left. Ricardo, our friend from Portugal, he's a professor of microbiology there. He writes, again and again, you do it over and over. Your podcast is everything about teaching. I'm expecting every Monday morning for the download to finish so I can have my pleasure drive to Ponte de Lima, a secondary campus of the university. Episode 72 was really good. The CRISPR and the iRNA sequencing was very interesting. But another thing made me wish my entire group of students could listen to you for. During the last episode, I found myself thinking, look how these guys learn. You are all great teachers, but you are also great learners. During your 90-minute talk, we get questions, chains of events, summaries, research, Google. It's almost like a fight for knowledge. Sorry for my English and congratulations. And then he sent a link to some interesting software he heard about called Tableau Public, which is a neat application where...
It really is neat. I looked at their little video. It's very cool.
If you have some data and you want to make a visual of it, you go here and they make interesting pictures. So check that out. Thanks for that, Ricardo, and thanks for your email. Etienne writes, I was listening to TWiV 72 and it was mentioned that archaea only live in extreme environments, hot vents, salt lakes, and extremely low pH, for example. Though it is true that they are well known to be the dominant life in those environments, it has been shown that they can be found in much more mundane settings. They even seem to play a major role in the production of nitrites and nitrates from ammonia in topsoil. For example, here are 2 articles about DNA sequencing revealing the presence of archaea genes in topsoil. I thought you might find them interesting, and one of them is the predominance of archaea ammonia oxidizing prokaryotes in soil, and the other is another soil article. So yes, we were wrong.
And thank you for giving us the dirt on archaea.
He's getting warm now.
It's warming up. Exactly. Thanks, Etienne. And Cedric writes, hello, gang. I'm currently listening to TWiV 72, and just heard the discussion of viral infections in archaea. One of the questions that came up was how viruses could survive the extreme conditions that archaea inhabit. I agree with Alan's assertion that if archaea can make proteins that survive such extremes, viruses can too, but it is also worth pointing out that archaea do not only live in extreme environments. They tend to
I think somebody just told us that.
They tend to thrive in nutrient-poor ecosystems and extremes of pH, temperature, and other environmental parameters, but they're also found alongside bacteria and eukaryotes in soil and other mundane habitats. Mundane again, wow. They're thinking in parallel. Like bacteria, archaea also live inside other organisms, including humans. And as you know, this is an excellent environment for horizontal gene transfer, including virus-mediated transduction. I love the show and always enjoy the discussions. Keep up the good work. So there are 2 corrections for us. Thank you, Cedric, and thank you, Etienne. Archaea are everywhere. Well, maybe not everywhere, but not just in the extreme places. Bill writes, I took the liberty of contacting the makers of the shutter timer at StudioHut.com that allows a regular camera to become a time-lapse movie maker and suggested to them the possibility of becoming a sponsor for your show. So this has to do with the Vaccinia movies of Jeff Smith that we talked about a while ago. They made these wonderful time-lapse movies of the formation of a plaque. I don't know if you saw those, Michael. They're just
I didn't.
Fabulous. They're in Science a month or 2 ago. It's a real-time movie of a plaque forming, and it's beautiful because if you teach plaque formation, this is the way to do it. It's great. And they use time-lapse technology, and we were musing about how we'd like to do that. So Bill sent us a link to this company that makes a shutter timer. I can't help but think there must be many who listen to your show who work in labs and classrooms and know as little about making time-lapse movies as I did. I think you were to try one of these in your lab, you'd feel the same way about the backup solution. So, well, maybe Studio Hut would like to sponsor TWiV. So Studio had then sent us an email and said, we were contacted by an avid listener of your podcast, and he said that you talked about making time-lapse movies, and we want to let you know that we sell photo equipment that allows you to do time-lapse movies. Would you be interested in trying one out? So that's from Mickey at StudioHut.com. So the time-lapse is great. You also need to have an incubator on your microscope. You can't just have the time-lapse. I don't have that yet, but we're working on it, and when we do, we'll definitely try it out.
I'd like to see whether polio plaques grow and look the same as vaccinia plaques
I'd
grow.
love to.
That would be a very cool experiment. But I not to get us out of a potential sponsor, because it looks like this is a very cool product for a lot of applications. But by the time you get the incubator and microscope set up, usually you buy that from a manufacturer that includes a whole turnkey solution, which comes with software that has the time-lapse feature in it.
Yeah.
So that's done that way now. But if you are looking for more of a hardware solution, then this Studio Hut thing looks like a very good arrangement.
Well, I think it's good of Studio Hut to be a sponsor because we have a virology audience.
Yes.
We have thousands and thousands of listeners who would probably love to hear about it, and they've already got a plug, so we'll pursue that. Duncan writes from Australia, hey guys, great podcast. Thanks. I'm plowing through the back catalog of episodes and listening to the current ones that are coming out. I'm not a virologist. Heck, I'm not even a doctor. But I'm finding the detailed discussions that you have fascinating, and it's really teaching me a lot of stuff that I'm finding interesting. In a recent episode, you were discussing the CRISPR defense mechanism that allows microbes to withstand viral predation. It got me thinking, if bacteria are susceptible to some viruses, is there any research underway into using novel viruses to attack active bacterial infections in humans as an alternative to antibiotics?
There was a fellow named Fleming who was working on that before he discovered something else.
There's also
De Harel originally. I mean, that was one of his original deals, was he wanted to use it to treat dysentery. This idea has been kicking for as long as bacteriophage have been around.
And it's not a dead idea. There are still folks looking into this, engineering bacteriophages that could persist, and actually wipe out the bacteria. The problem, of course, is that you put a bacteriophage into a bacterium and it's replicating every 20 minutes or less, and they tend to evolve and mutate extremely fast, and you lose control of them very rapidly, and they achieve a balance with the bacteria, which is not quite what you want.
I actually have right here on my desk a beaker that has in it 3 bifurcated needles for smallpox vaccination that I use for demonstration, and a vial of phage that's supposed to attack an intestinal bacteria that was brought back to me from Russia by Donna Duckworth. That's got a bunch of Russian writing on it, or some other sort of writing, and it says intestiphage. And so I'm just waiting to get sick enough to chug this.
Yeah, they have a big program in Russia, I heard, using phages. And in fact, the book Arrowsmith was about a fictitious virologist who was trying to use phage to treat bacterial infections. One of the problems is they're very antigenic, and when you inject them, you make antibodies against them rather rapidly. But yes, it is being considered. And let me do one more quick one. Let's run down to Angie here, who says, hello, fellow virologists. Just wanted to drop you a line to say I love, love, love your podcast. I just discovered it last month, and I can't get enough of it. I'm a former virologist. I worked on herpes and parvovirus, who is now a forensic scientist. Although I love my new job, I miss reading and discussing the newest papers, research on viruses. This podcast fills that void. The way you discuss papers and answer email is great. It reminds me of journal clubs or informal discussions at lunch or conferences. Thanks, and keep the podcasts coming. They are infectious. P.S. Remind your listeners, if they like an article you discuss but can't access it, they can always email the author and request a PDF copy of the article of interest. Over the years, I have done this many times, and no one has ever refused my request. Most will even answer questions about the article. A little hint, emails to the first authors usually get answered more quickly and with more enthusiasm than emails to the last author.
And that's not just true of paper requests.
That is true. I must say that's good suggestion there, Angie. Thanks for that. Let's do a couple of science picks of the week. Michael, you still with us?
I am. A couple more minutes, and then I have to get ready for my 3:30 meeting.
All right. Well, you can interrupt us at any point. We'll say goodbye.
Okay.
Rich, what have you got today?
So last week there was a frontline show on PBS called The Vaccine War that can be accessed online, and I actually watched the online version of it last night. And I think it's very frontline. You know, it's what I thought of was a very balanced presentation of the controversy over vaccine, and it interviews people on both sides of the equation: Anthony Fauci, Paul Offit, Jenny McCarthy, a bunch of notables from the CDC and HHS, and highlights a community in Oregon where there's a fairly substantial fraction of the population that are not vaccinating their children because of fears about vaccines. And, just kind of reports this stuff and leaves you to draw your own conclusions. And I just thought it was very well done and done in a very understandable fashion. Now, of course, if you read the comments that people write on the show, they're as polarized as the debate itself. But I think the show's worth watching to inform yourself on it. I'm very interested in this place. It's in Ashland, Oregon. I think that was it. And yes, Ashland, Oregon. There's a large percentage of the population there that are not vaccinating their kids, and so the way I see it, that's an experiment. It'll be interesting to see what happens there.
Yeah, this is a good program.
So it's a good show.
Everyone needs to look at this. You could watch it online, I guess, or do you just...
Yeah, I watched it online last night. Piece of cake. Very easy.
All right, good. That's a good one. Yeah. All right, Alan.
Mine is a very little piece of software called Readability. And if you go to the website for this thing, you can take the little gray tag that says Readability on it and drag it up to your browser toolbar and drop it. It's a bookmarklet. It's a little piece of JavaScript. And what it does is next time you're visiting a web page, like a journal article, for example, that you're going to be reading an extended length of text, you hit this little bookmarklet in your toolbar, and it reformats the web page for you.
Ah, this is beautiful.
Right in your browser, and it does it by proper typographic rules, which so few web designers understand. It puts it as a single column of text, approximately 70 characters wide. It's laid out appropriately. It's black on white. It's in a serif font that's easy for extended reading. It's appropriately sized. It makes it readable, which is why it's called readability. And I found this thing. A friend of mine turned me onto it a few days ago. I'm totally addicted to it, and think it should be a default option on all browsers now. So if you do extensive reading online, and you go to a website and there's some long article you want to read, but the text goes on in massively long lines of fine print, which commonly happens, then you just hit this tab and all is right with the world.
I'll be darned. That's amazing.
Huh.
Yeah.
This is great. No, reading on a online page on a computer screen is difficult because it's not formatted right. This looks great. Right. One morning when Gregor woke from troubled dreams, he found himself transformed in his bed into a horrible vermin. Now, what would that be from? Isn't that Kafka?
Metamorphosis, right.
Metamorphosis. Yep. Great. Thanks for that, Alan. Well, my pick is a book. It's a little bit of a fun jaunt. It's a science fiction book by an author called Jerry Pournelle, Pournelle. And it's a small book about a boy living on a foreign planet, or a planet, not Earth, of some sort. He has an artificial intelligence chip implanted into his head. And he goes through school being told all the answers by a computer, a remote computer that can talk to him. It's very interesting. But on this planet, it's very interesting. Earth has colonized it, and there is a conflict going on. But the planet happens to be inhabited by a neural network organism who eventually plugs into this little boy's brain. It is so interesting. There happen to be centaurs that live on this planet. So I think it's a lot of fun. It's short. You can read it in a couple of hours, and it's really cool. And if you want to have fun, we give you a lot of picks that make you work hard. But if you want to have a good read, check out Star Swarm. You want to go, Michael?
Yeah, I need to get going.
Thanks for joining us, Michael. Appreciate it.
Yeah, thanks very much. It's great talking with you.
Great
Thank
talking
you for
to you.
inviting me.
All right, we'll probably get questions, and if you don't mind, we'll send them along.
Yep, sounds good.
Take
All
care.
right.
Bye-bye.
Bye-bye.
All right, let's wrap up TWiV. We've had a lot of new listeners coming to TWiV lately, and if you are a new joiner, consider subscribing either in iTunes or at the Zune Marketplace or by the RSS feed so you automatically get new episodes as they come. If you used iTunes, go leave a comment there. It always helps keep us on the front page of the Science and Medicine directory. TWiV is part of sciencepodcasters.org, promednetwork.com, and microbeworld.org, websites where you can find great science podcasts and other science information. As always, send us your questions and comments to TWiV at twiv.tv. What do you think, gentlemen? Did you like hepatitis B?
Well, I love it. I like these episodes that have an expert on to talk about, spend the whole time talking about one virus that we haven't talked about much before. And I had a lot of questions about HBV that had accumulated in my mind over time, and it was nice to have an opportunity to ask somebody about them who knows what's going on.
Yeah, I also like these. It's a lot like my regular work where I talk to researchers on the phone and discuss what they're saying. But really fun to have somebody dive deeply into a topic like this.
It's good when you have a couple of people, like instead of one-on-one as you usually do, right? You have a conversation, and that just adds, I think for listeners, it adds a lot, right?
Yes,
absolutely.
I agree.
But you guys think of things that I don't and so forth, and I think it ends up being a very enjoyable conversation.
I think for a listener, it's even nice just to hear different voices.
Yeah, absolutely. Right. I would like to do this more often. It's just a matter of scheduling, It's not our primary job, so scheduling.
People must be knocking down your door wanting to be on TWiV, Vince.
Not really, no. But when I ask, they usually say yes.
I
don't have too many people refusing. But, it's funny. A lot of scientists are not up on the Skype business. He happened to have already used it, but a lot of people don't have it, and then the quality is not very good. So it takes a little bit of work, but I'm willing to do it because I learn a heck of a lot, too. I think
it's great.
Anyway, thanks for joining us, guys. Alan, thank you.
Always a pleasure.
Alan's at dovedocs.com in Western MA. If you're ever wandering through the forests of Western Massachusetts, you'll see him there planting his garden.
That's right.
And Rich Condit, good to have you back.
Great to be here. Always a good time.
We are going to be in we're going to be in Florida soon, next month, May.
Yep, pretty soon.
May is just around the corner.
Yep, May is this weekend.
Yeah, tomorrow, right?
There? Yeah.
Well, we're going to do a TWiV from the University of Florida at Gainesville, which is in north central Florida. I'm going to go down there. I'm going to bring my equipment, and we're going to record with a couple of virologists, the same kind of thing you've always been used to, and that'll be a lot of fun.
Yeah, we're looking forward to it.
You've been listening to This Week in Virology. Thanks for joining us, everyone. We'll be back next week. Another TWiV is viral.